Hello everyone, welcome to video 12-2. Today we're going to be looking at how the immune system defends the body against germs and creates an immunological memory. And we're going to do that by looking at how viruses infect cells. Here we're looking at a bacterium called a bacterium that's infected by these viruses called bacteriophages. And viruses all contain the following things. The structure of a virus contains nucleic acid like DNA or RNA and a protein capsid. Sometimes the virus may also have an envelope which is made of a phospholipid bilayer which is stolen by the host cell, and it also may have these proteins which allow the virus to infect cells, but not all viruses have those outer parts. So here we're looking at a picture of a virus that's infecting a cell. Here's the virus that's landing on the cell and injecting its genetic information, and the cell thinks that that information needs to be transcribed and translated. So proteins are ultimately going to be made, the viral capsids specifically, and that allows the virus then to emerge out of the host cell to infect new cells. This is known as the lytic cycle because what the virus is doing is it's bursting out or lysing the cell. Viruses can do something even more insidious than that, and that is called the lysogenic cycle. The lysogenic cycle is when the virus inserts its DNA into the host, such as shown right here, and then that virus information, the DNA, is embedded into the genome of the host cell. So when the cell then undergoes DNA replication, the viral DNA is copied. And so the descendant cells actually have that viral genetic information embedded into their genome, and as the host cell reproduces, more viral DNA is passed on to descendant cells. And that information of the virus is like a ticking time bomb. So when the virus then can later emerge, going from lysogenic to lytic, it can destroy the host cell and causing lysis. So let's look at why must viruses infect cells in order to reproduce. So recall that viruses don't have ribosomes, and so in order to make more virus, you need proteins. Proteins, of course, are made at ribosomes, which viruses lack. Next thing is, why do some people get shingles years after contracting chickenpox? You may have seen some of those commercials depicting older people getting shingles, which is really chickenpox later in life. And so when I was a kid, I had chickenpox when I was 6 years old, and then years later on, when I was in graduate school, I got these sores or pustules on my skin, which I went to the doctor. He told me I had shingles. And so what I didn't realize is that the virus that causes chickenpox was actually hiding in the neurons of my spinal cord. And so all those years later on, the virus was able to reemerge. Usually that happens if a person's immune system has been lowered, if you're under stress and not getting enough sleep. And then the virus senses that weakened immune system and then reemerges, coming down the spinal cord or coming from the spinal cord out of the nerves and then out of the skin, creating those pustules. So someone that has never had chickenpox, if they come into contact with someone with shingles, they can get chickenpox from their shingles. Next thing is, why are antibiotics effective against viruses? And the answer to that question is no. So recall that when we use something like penicillin, what that's going to do is it's going to interfere with the bacteria's peptidoglycan cell wall, and that prevents the bacteria from making more cells. Other types of antibiotics like streptomycin and tetracycline, they inhibit the ribosomes. And of course, viruses don't have ribosomes. So antibiotics are completely useless against viruses. So next time you're sick with, let's say, the flu, don't ask for antibiotics, since that would not be effective against the influenza virus. So now let's look at how vaccinations work to prevent someone from contracting a disease. So let's say you're exposed to an antigen from a germ, like this pathogen here, which might be a bacterium or a virus. Recall that antigens are the cell surface marker proteins on the host cell, which are like keys which allow it to attach to a target cell or target tissue. And the word antigen comes from antibody generator. So ultimately those antigens that are on the pathogen will help your B cells to generate antibodies, as we'll see. So it's going to take you time to figure out what type of antibodies to make to that germ. So during that period before you figure out what type of antibodies to make, the pathogen is causing damage to cells. For example, a virus would be lysing cells, causing inflammation, which causes a fever, swelling, all kinds of bad things, and you're going to feel really miserable during that time period. So after a while, your B cells hopefully do get activated, and you're going to start to generate those B cells, those antibodies from those B cells. And that's what's being depicted here. So this is the amount of antibodies that are being made in the person's blood. So if you're exposed to the pathogen a second time, this time you're able to recognize that germ much more quickly. Instead of having to wait maybe several days to figure out what type of antibodies to make, you have this ability to remember the germ and generate huge amounts of antibodies in a relatively short amount of time, like within hours. And that would immobilize the germ and then preventing it from causing any kind of damage to cells and therefore inflammation, such as fever. So what we call this is immunological memory, the ability of the immune system to remember a pathogen, and that leads to this rapid immobilization of the germ, stopping the germ from causing damage to the cells. So what vaccines do is vaccines are weakened or destroyed pathogens which are used to stimulate immunological memory. And so you can give someone a vaccine. You can imagine like that would be the first exposure, but instead of actually the germ, it's just the destroyed or weakened germ. And what you're really giving them is the antigens, not the actual virus or the bacterium. And so by giving them the antigens, that's going to stimulate B cells to make antibodies. So if you ever get really exposed to the germ later on, you have that immunological memory to create those antibodies very quickly. The first person to use a vaccine was a British physician named Edward Jenner back in the late 1700s. And what he noticed is that milkmaids, women that would get milk from cows, would sometimes get these pustules on their skin, which was known as cowpox. And these women never seemed to ever get smallpox. And so Jenner hypothesized that maybe the cowpox was somehow giving these women some type of an immunity to smallpox, a really deadly disease that killed thousands and thousands of people every year around the world. So he inoculated a boy with a little bit of cowpox serum and then exposed the boy to smallpox. And of course, the boy never got smallpox. He never died. And so by the early 1800s, from which this cartoon was made, more and more people are getting vaccinated with cowpox. But for some, that was seemed to be going against nature, and that there was this fear of taking something from a cow or a cowpox and mixing that with people. And so there was concerns about that for some. And today we have our own controversies. So there's some people believe that vaccinations cause autism, which was based on a discounted paper from decades ago. But some people continue to believe that idea. And so the sad thing about that is that some people are now not getting vaccinated, and so the rates of measles, which is something that was really not seen very much for decades, is now starting to reemerge. Like in 2017, there was a large outbreak of measles in Minnesota. So next idea, let's look at how allergens work. You may recall from the last video that allergens are going to irritate the sinuses, where there is a type of cells called mast cells. Those are in the dermis of your skin, such as in the epithelial tissues of the sinuses. And then when those mast cells get damaged or irritated, they release histamine, that chemical alarm signal that's going to trigger inflammation, and that leads to swelling and a fever and so forth. So one way to prevent that from happening is to take an antihistamine, and that would suppress that inflammatory response, so you wouldn't have the symptoms of inflammation. Another topic we're going to look at is how does HIV work. So HIV stands for Human Immunodeficiency Virus. And here we're looking at, this is the capsid of the virus, and here we have these the cell surface marker proteins which allow it to attach to a cell. And so viruses, well, specifically HIV rather, comes from actually monkeys and apes. So in Africa, people consume wild animals as they do, eating deer around here. But the thing is that we are also primates. So if you were to, let's say, butcher a monkey or an ape, potentially some of the blood from that animal can get into your blood, and viruses might be able to jump species. And that's what happened in these two separate cases of people likely slaughtering a sooty mangabey and getting this form of this virus. So it's called SIV, or Simian Immunodeficiency Virus, which jumped species into humans, and then later on that happened with another form of virus from chimpanzees. And that started in the 1920s, and now that HIV is spread into people. So HIV is fairly insidious because what it's going to do, it's going to wipe out your immune system, the body's defense system. And so here we're looking at HIV, and so it's an RNA-based virus, and it's going to inject itself into the host cell, and you can see it has this RNA. In order to hide lysogenically in a cell, it has to copy its RNA into DNA, and it's going to use an enzyme called reverse transcription, or reverse transcriptase, to do reverse transcription, because normally you recall that transcription is when you go from DNA into RNA, like mRNA. But here, with the virus starting with RNA, it has to go backward through transcription, and this is called reverse transcription, and uses an enzyme to do that. Once that has happened, again, it can hide lysogenically in the host cell, and the person could be HIV positive for years and years and years without actually really having AIDS. We call these types of viruses retroviruses. Retro means backward, and so the virus is in a way going backward through a step. Instead of forward through transcription from DNA to RNA, it's going from RNA to DNA, sort of a backward step. So they're called, these viruses that are RNA based are known as retroviruses because they undergo reverse transcription. Once the virus emerges from the host cell like that, further cells can then be infected, and then the T cells are wiped out, and so the person's immune system is greatly diminished. Last thing is, how does someone die of AIDS, acquired immunodeficiency syndrome? Well, you can imagine if your immune system is highly compromised and deficient, it would not be that difficult for maybe the common cold or the influenza virus, which causes the flu, to infect and potentially kill that person. And so people often die of common diseases as a result of AIDS. All right, thanks for watching the video, everyone.